Hello everyone, welcome to week 8 session 5 of NPTEL course in Herbal Product Technology and Regulatory Sciences. This week we are seeing the quality control methods, especially with regards to the regulatory requirements. So we saw what is pharmacopoeias, we saw WHO, ICH as well as Ayush guidelines. In this session we will differ slightly from the previous 4 sessions in that we are going to see definitely quality control methods, but we are going to be using a unique markers which are called as molecular markers. So here in this particular session we will see the role of molecular markers in quality control of herbal drugs as well as formulation. So what are molecular markers? Molecular markers may be markers which are obtained from enzymes as well as obtained from the genetic material. Particularly in this session, we will see the molecular markers obtained from the nucleic material, and that is your DNA based markers. So have you heard of this thing called as paternity testing or DNA testing, or maybe you might have seen movies in which or some series in which they particularly pick up DNA samples and use it for analysis. So in this particular session we are going to use our own magnifying lenses, check in the DNA and try to analyze different herbal samples, formulations, and how can the DNA of a herb help us in quality control. Let's understand that. To start with, whenever you have a plant cell, inside the plant cell you have genetic matter, and this genetic matter is made up of DNA, and this DNA can be in nuclear or it could be a ribosomal DNA matter, or sometimes this nucleic matter comes out of nucleus in form of what is called as transcription, and then later on goes in the process of translation to form proteins. So your proteins as well as enzymes come in from the genetic material. So this genetic material differs not greatly, very slightly, but considering the number of base pairs that are there in DNA, they can help us in identification. So in this particular session, let's get into our DNA fingerprinting. So what is DNA fingerprinting? DNA fingerprinting is a technique where you create a fingerprint for a particular organism. In this case, let's create a fingerprint for a plant. We know our DNA is made up of what you call it as a sugar backbone, as well as there are something called as base pairs. These base pairs are your adenine, guanine, cytosine, thymine. Now this base pairs are something we are going to be looking for when it comes to diversity. So plants in the species also slightly differ in their DNA. This unique differences in DNA can be used as markers. So DNA fingerprinting is a technique that uses DNA markers which rely on a subtle difference between species or the base pairing in the sequencing of the nucleotide bases to create what is called as a diversity or a species or a variant. Now this is picked up, this differences is used as what is called as determinant, or this differences or the region which gives this differences are called as DNA markers. Now DNA markers have been used to identify plant cultivated varieties. They are used to assess genetic diversity as well as to confirm ownership. Now where does ownership come in? Now with your plant trade going globally, there are certain agreements like your Biodiversity Act, there are certain agreements like your TRIPS agreement wherein geographically you can register your plant as a geographical indication, and that to prevent the use or abuse of that plant. So if you have a unique DNA fingerprint to your plant, you can identify your plant and nobody else or no other country can put their claim on that particular plant species. So that's how your DNA markers help. Now, how are they going to help us in this particular session or in this particular course is, you can use the very same DNA fingerprint to understand if this herb has been added to the formulation, or instead of this herb some other herb has been added, because in that case that would definitely be missing the genetic diversity, or that would definitely be missing the DNA markers that we are looking for. So, DNA markers are basically genetic markers that are very specific for a plant species, that are identifiable by their base pairings and inheritable. So, from the plant to the offspring they will carry, and that is how you have your paternity test. So, very much same in the plant family as well. You will see them, the offsprings will inherit the parent DNA, and with known chromosomal location they will help us track these genes, assess variation and identify the plants. So, let us go little deep into it. How do you use and what are these techniques? So, if you want to check the DNA fingerprinting, or if you want to carry out DNA fingerprinting, the process is very simple. You have to take genetic material out of the plant. How do you take it? Take any sample, say for example your leaves, your stems, sometimes something like your pollens, anthers, any plant sample you can take, and that should be sufficient to give you the genetic material, that is your DNA. So, how do you extract the nuclear material outside the cell? The first thing is, in order to keep it intact, people sometimes freeze the sample and then mill it, or you could coarse grind it for a simple extraction. So, grinding step to rupture the cell walls is done, and post grinding you do the centrifugation. So, when you do the centrifugation, what happens? Most of your plant debris, when you dissolve it in water buffer with a little surfactant called as CTAB, that is cetyl trimethyl ammonium bromide. So, this you are going to extract it mostly in water, usually a buffer containing surfactants such as CTAB and containing chelating agent to stabilize your DNA, and other such materials you will add it. And what will happen when you centrifuge it? The plant material will go at the bottom and your DNA will remain suspended in the dissolved form. So, now that your DNA is dissolved, you centrifuge and remove the plant debris, and then you can slowly precipitate the DNA. The DNA can simply be precipitated by adding any low molecular weight alcohol like ethanol, methanol, which renders it inactive, as you can see here in this. This is the little DNA. So, from that clear supernatant you could easily remove. Sometimes nowadays even you have pre-preparation steps as well as filters which are available, which will give you this kind of clarified solution, and then this DNA is precipitated by addition of alcohol. Now, once this DNA is precipitated, the DNA is very long, thousands and thousands, in fact 10 raised to 23 base pairs or so, depending upon the plant species. So, now this DNA, if you have to assess, you to cut it. Sometimes this is cut with the help of specific molecular reagents, and sometimes it is cut with the help of enzymes. These enzymes include what are called as restriction endonucleases. Now, restriction endonucleases identify a particular region, a base pair combination, and then selectively cut. Now, when they cut it, what is going to happen? You are going to get the fragments of DNA. Now, these fragments, depending upon where all the cuts are produced, where all the restriction endonucleases bind and can produce cut, you can get different cuts, because say for example, this restriction endonucleases is picking up ATG, ATG. It is not everywhere that you will find ATG, but wherever it is going to cut in that region. So, say for example, it cut at 5-6 places, I am going to get some fragments, and this location of ATG will not be same in all the species, and as a result I get different fragments. So, if you carry out something called as gel electrophoresis, using 1.5% agarose to create a gel or page electrophoresis. So, you will see what is called as agarose electrophoresis mostly used for this technique, and then you can separate it. So, electrophoresis will use a positively charged electrode, and what happens is your DNA, because it is negatively charged, will travel here. The larger pieces, you can see 1000 base pairs, will remain back because they are fat and they do not diffuse through the meshwork of the polymer, whereas your smaller pieces like your 100 base pairs will travel ahead. So, you will get a good gradation of pieces in terms of size, and that gradation is unique to every plant. So, you can see sample A has gotten this fragments after cutting with this, sample B has got a different set of fragments, so does sample C. So, now what happens, if you have to check your sample A in the formulation, say for example, this is mango, this is apple and this is banana. So, what is going to happen tomorrow if I come across a same pattern as that of mango, I definitely know that this is also a mango ka DNA. Now, when I am doing this, at the same time, if somebody claims it to be banana and if I get this pattern, I can definitely identify and say that the given sample is not banana. So, that is how your DNA fingerprinting helps you in authentication, and then you can use DNA markers for it. So, what are these DNA markers then? Now, DNA markers are unique DNA sequences, as I said, and that DNA markers, depending upon what they are, you can have different techniques that are involved. This DNA markers can be single nucleotide polymorphism. What is single nucleotide polymorphism is, instead of AUG in mango, if it is ATG in banana, what is happening? It just one thing that is, or one nucleotide that is changing. So, SNPs are more of what is called as single nucleotide polymorphism. You also have what is called as short tandem repeats, variable number tandem repeats. Another markers are restriction fragment length polymorphism. You have techniques such as these are basically the markers, short tandem repeats and VNTRs. What are STRs and VNTRs are? So, I will just give you one example. Say for example, this is your DNA material, okay, and in the DNA material, say for example, I am picking up this whole sequence as 1. So, what I see is AUG continuously keeps on repeating. Now, this keeps on repeating, say for example 50 times. So, because this is repeating 50 times, I am getting AUG about 50 times here. Okay, so maybe about 50 lines or so. So, what happens is this region is a AUG short tandem repeat. So, short sequence but many repeats. So, what happens is one species it might have 50 repeats, in another species it might have 200 repeats. So, number of repeats for a particular sequence changes, and that is what is referred to as short tandem repeats. Now, coming to VNTRs or variable number tandem repeats. Now, imagine if this was a big sequence. Maybe 25 base pairs or so, okay, and then this bigger sequence keeps on repeating. Now, short tandem repeats generally consist of a small combination, variable number tandem repeats is a larger combination, and that also keeps on changing depending upon the plant species. So, you could use this also as markers. Now, you can use other techniques also which uses what are called as restriction fragment length polymorphism, the one I discussed, where your restriction endonucleases produce different fragments. Then you have what is called as RAPD, we will just see it, SCAR, that is sequence characterized amplified regions, and amplified fragment length polymorphism. To understand this, let us see it diagrammatically. Now, when I am talking about single nucleotide polymorphism, is the first case So what is happening is here throughout the DNA remains the same, but at one particular base pair between two plants it is differing. Or say for example between Alfonso mango and between Kesar mango this is differing. So I can use this as an identifying trait to differentiate between Alfonso mango and Kesar mango. Similarly, as discussed, you can see here there are STRs, that is short tandem repeats, and here there are VNTRs, little bigger ones, and you can again cut it with help of molecular scissors. We have done Cas CRISPR studies or maybe restriction endonucleases based studies, and what you do or what you get in the end is this regions. So how many such regions are repeated you come to know, and that can help you identifying the plant. Going to RFLP, like I said, restriction endonucleases are used and you cut your DNA. So we saw here you can cut your DNA and you can see your banana versus mango. So imagine using the same technique here. So you know that this is one of the parent, say father, this is your mother, and generally the offspring gets half of the genetic material. So the offspring is mother plus father, that is how it is done. So for plants also similar traits are checked, and then you can sum it up for a particular offspring as well as you can compare it with other plant species. Now going to little advanced techniques, we use randomly amplified polymorphic regions, and that uses what is called as primers. Now what are primers? Primers are regions which stick to a particular region of DNA. Say for example, when I am using a DNA, and suppose if I am able to open it, this is done via a process called as polymerase chain reaction. So in polymerase chain reaction, first using a heat or a higher temperature like 90 degrees Celsius, the strands are kind of made open. Now once the strands are made open, you stick what is called as primer to it. So say for example this is a strand and here you stick a primer to it. Now what will happen? Because of the polymerase that you are adding in a polymerase chain reaction, it will go on adding nucleotide base pairs and this will go on preparing. So wherever it has broken, now from there it will start synthesizing complementary pair. Now this process keeps on continuing till you have very much enriched fractions of these fragments, and these fragments are the ones which are used to what is called as determine the identity. A little advanced technique is what is called as AFLP, amplified fragment length polymorphism. In amplified fragment length polymorphism, whenever the cutting happens, imagine the cutting is happening here. So this is the complementary pair. Let's prepare a complementary pair. So GC, GC, AT, TA. Okay. Now say for example your enzyme is cutting it from here. So what is going to happen is, because of its restriction enzyme, it is giving what is called as asymmetric cut. So now your DNA is cut from here. So now what you have is here a strand which is ATG. We call it as a overhang. Now if this is annealed or broken by heat, you will get a single strand with a ATG. Now imagine you stick a primer to it. Okay, you stick C, you stick A, you stick T, because it's a complementary and your enzyme is used for that. You will get an alternative strand being prepared. But because it is digested by the same enzyme, the other side also. So now other side is also complementary and this keeps on multiplying. So it's just your RAPD in an amplified version and you get what is called as amplified fragment length polymorphism. It is basically when two primers are used at both the ends and you get a good segment formation amplify it much more time, you will get a good single fragment enhancement. So that are few techniques which I use. This are, I mean, I'm just being brief, but this is the way it's generally done. So in addition to that, imagine if in your AFLP, your DNA material, what got characterized as your ALP fragment or AFLP fragment is nicely genetically coded, and you know all the base pairs of this. So this is just a continuation of AFLP, and that is what we are going to use in our session today. So genetic techniques are more given in biotechnology lectures, and you can also use some YouTube videos, links that we'll be sharing to understand these techniques in depth. But understand, in AFLP, if you take this fragment and characterize the sequence of this DNA fragment, so in that case, what you get typically is what is called as sequence characterized amplified region. So SCAR stands for sequence characterized amplified region, where the region where you have to, which you have randomly amplified within what is called as the flanking regions or the cutting regions using RAPD technique, that is rapidly amplified polymorphic domain region, that is something which is going to be amplified. And if the sequence of this DNA is known for your plant, you can easily use it for markers. Now this sequence is well studied. How do you do it? Say for example, again take a plant, extract the genetic material, use the genetic material, do what is called as the DNA fragment separation. When your fragments separate, apply a what is called as primer, as we saw to it, and create a strand. Now this strand is a rapidly amplified region. Now take this rapidly amplified region and do a genetic sequencing or do a DNA sequencing, especially for the base pairs. Once all the base pairs of this are known, it is called as sequence characterized amplified region. And once you know the sequence of all of this, what you can do is you can pick up a particular region which is unique to plant. So pick up this region, maybe say for example this region is unique to Vinca rosea. So I know that because this region is unique to Vinca rosea, next time I will target this region. How? I will produce a complementary strand. So say for example this is the SCAR. SCAR markers are generally very big, but I am just giving you a small region. So the SCAR markers, take for example for a particular herb is TGCA, TGAT. Okay. Now I stick a small primer. I know this small primer, let it be like A. Okay. Now when I put this primer and then I put what you do or what is called as a PCR, that is a polymerase chain reaction, a complementary strand is completely produced. Okay. So then because of PCR, I again get a whole strand. Now when every cycle of polymerase chain reaction happens, this get amplified like raised to n. So you will have a very enriched region in this. Now next time when I have to study or check does my formulation really contain Vinca or not, I just will use this particular primer which is radio labeled. Now once I radio label it, I know in my electrophoresis, if this is the primer band and this is the sequence which I am getting for my Vinca, because they are same and same in size, that means this is a Vinca sample. Tomorrow even if I get a formulation which has Vinca, I will do the same thing. From that formulation I will extract the DNA, I will use a polymerase chain reaction using this as a known primer, and then I will check from that sample DNA is anything binding to this radio label. So if it is binding, that means my formulation also has Vinca sample. So this is used to identify polymorphism. Tomorrow if there is another Vinca variant which is not required, but that doesn't have this sequence, I know that it is other vinca variant and not the one that I want. Say for example, between white vinca and pink vinca. This is the region which varies. The primer will not bind to pink vinca, it only binds to white vinca. So I can even understand whether the white vinca has been added or my pink vinca has been added. In case of pink, it will be missing. So here again, SCR techniques generally use 20 to 25 base pairs. They can be used to identify target species even from a pool of related species and produce a very bright band, because only that region is amplified in PCR, because other regions, they are not having the primers, they will not get amplified in your PCR, and SCR markers have been used to identify Artemisia, Phyllanthus, Panax, Echinacea, Vinca, Rauwolfia and many other species using the same technique which I mentioned here. Now we go on to other places where it is used in formulation. It is used even for determination of amla, wherein a SCR markers was developed with as little as 0.1 gram sample. Now this was amplified, only that region will get amplified, and then when you check amla churnas or triphala churnas by extracting the DNA, you should get the same band. So this is your, say for example, SCR radioactive band for amla. If I check my triphala churna, I should get the same band if amla is present. If I get amla churna, I should get the same band. If this band is missing, that means amla has not been added to my sample. Now moving on to another agents or other particular domains which have been used. So this regions or DNA barcoding regions include your nuclear genomic regions as well as chloroplast genomic regions. So nuclear genomic regions are more pertaining to the nuclear DNA, and this includes your ITS, especially your ITS 1 and 2, whereas chloroplast genome includes the what is called as ribosomal DNA, which has been used, and the marker regions which are used for checking the variability between the plants include your MATK or MATK, RBCL, RPOB, RPOC and so on. So what are these names and how do we come across this? Let's try to understand them. So DNA barcoding using RBCL as well as maturase or MATK has been approved by consortium of your barcode of life. Now this is basically a working group which documents the DNA fingerprinting of most of these plants, and they recommend using these two barcodes because they see that as the plants evolve, different genomic regions remain similar, some genomic regions start showing diversity, and these are the ones they identify which are showing diversity as the species of the plants varies. So when you have a phylogenetic tree like your angiosperms, gymnosperms, within the angiosperms it was formed that your MATK as well as your RBCL are the ones which can really be used. So what is this RBCL and what is this MATK? RBCL is oftenly referred to as Rubisco-Large subunit region. It's a very versatile, very diverse region, very easy to identify, and because you know the primers of it, it is used to give a interspecific or interspecies identification. But its resolution is very low. So it is one more set of identifying domain is used, and that is your maturase K. And maturase K has higher rate of what you call it as resolving power even at species and subspecies level. So what is this RBCL? Now RBCL is actually a gene on a DNA that is almost 50 to 55 kilodalton large. It's basically part of a large subunit of a coenzyme which is called as Rubisco coenzyme, or often called as your ribulose 1,5 biphosphate carboxylase or oxygenase enzyme, which is used during the photosynthesis for CO2 fixation. Now this is located in chloroplast or cpDNA. Now from this, the Rubisco region which codes for this enzyme is picked up because the starting sequence is same, but as the sequence, that is your 50 to 55 kilodalton sequence goes on, you will see number of base pairs, and within the species this base pairs vary. Though the initial sequences may be the same, in between there is difference, and this difference is picked up for barcoding. Similarly, you have maturase K. Maturase K is more of what is called as a plastid region, and it is located within a intron. Intron is something which stays inside the nucleus, whereas exons go out for translation process. So, it is an intronic region. It is known to induce splicing, or what is called as breaking up of DNA. So, it is situated in chloroplast genome, especially within what is called as the tRNK region, and it is coding for about 1500 base pairs. Now, within this 1500 base pairs, some base pairs may vary as per the species, and that is again what we use for. So, if you see a good example of this I can give you is your herb, that is your momordica, that is your Karela or a bitter gourd. So, momordica has numerous species. So, you can see here there are different species, and I have not put all those 1500 base pairs, but few base pairs that shows variation. So, the number at which they occur is all given here, and you can see here that within that 1500 base pair there is diversity. So, you have momordica balsamica, but even if you see here this region which is momordica charantia, within that also there is a good variation. So, this variant regions have been picked up, and you know that this, say for example here within your momordica dioica, momordica charantia, you will see your momordica sublanguta is slightly different as it has A instead of a C. So, you can use this base pairs, and now that I know the initial part, that is 1 to 200 may be the same part, but because there is a diversity here, I can use this diversity for my own advantage to code for it. So, this is how the domains or differences vary. The sequences are picked up, the sequences are characterized, and only specific sequences are characterized. You need not characterize the whole thing. You just need to characterize 1500 base pairs, and now once you know all this 1500 base pairs, you just quickly read this ones, and depending upon what is the sequence at 566, 568, depending upon what is the sequence at 1304, 1303, I can tell you which species of momordica has been used in my formulation. Similarly, I can use a blend of it. So, say for example, I can use a primer sequence. Like I said, it is the one which is used to bind. For maturase as well as RBCL, this was used for identifying bamboo samples, and it was seen that whenever I use this sequence in combination, I get still better results. So, for bamboo samples, you can see here, first one is just a ladder to identify the molecular weight. Second are genetic samples only identified with MATK. Third and your third and fourth are the ones which have been identified with RBCL, and all of them gives you a very close, nice, unique amplification. Why? Because the other regions in bamboo, because they do not bind to that primer, do not get amplified, and you can get a good radioactivity, or you can stain it with ethidium bromide and get a good staining. Similarly, for your nuclear DNA, you can use what is called as internal transcribe spacer, which is also a highly variable region located in your what is called as the ribosomal DNA or rDNA. It is located somewhere between your 18S and 5' region of your ribosomal DNA, especially your subunits of DNA. Now, this also it is seen that compared to your RBCL or your maturase, ITS has still better interspecific resolution, and that is also used in standardization of formulations. So, if you go to see its application, there is a plant in Ayurveda referred to as Gambhari. Okay. So, Gambhari, it is your Melina arborea, and it is often adulterated with other species of Melina or even with Monotus nordifolius. So, here what you see is when you use this primer sequence, generally for your Gambhari, which is a genuine sample, the 142 base pair should be amplified. But what happens is instead of Gimellia arborea, if you added Gimellia asiatica, Instead of 140 base pairs, your primer is now only 93. Go back to here. What does 93 mean to you? That means instead of this, your band will be something less. So it will go little ahead as far as the ladder is concerned. So this gives you a little heterogeneous response, and you know your arborea has not been added, your asiatica is added. Similarly, for your Malotus nudifolius, what you get here is instead of 142, you will have 150 base pairs. What does that imply? Again going back, your typical amplification you should look like that, but now because it is higher, it will give you somewhere a band at this particular position. So if this is 142, this is your 150 and this is the lower one. So depending upon the genetic size, you can easily identify the adulterants in plants. So I have given you paper links to identify this, but these are fewer techniques wherein your DNA based markers have been used. Now, you know, in terms of compounds, some compounds are present. So even if you take marker compounds may be present. But when you do it at a genetic level, you are very sure to the species, variants or even cultivars level. But yes, you have to weigh the techniques judiciously, as not all techniques would be able to identify species or subspecies level. It only depends on how you identify the genetic variation in that. So here are few references which can help you understand in detail how your DNA based molecular markers can help you identify adulterants, identify plants as well as authenticate them. With this, we finish our 4-week duration with you. Thank you everyone for your support as well as happy learning. Thank you.